In this episode of our cell culture series, we're building the foundation for everything that follows by understanding the fundamental differences between prokaryotic and eukaryotic cells. Before you can culture cells effectively, you need to understand how they are structured, how they grow, and how they function. From bacteria to mammalian cells, these biological differences determine culture conditions, growth requirements, media composition, and experimental design. Why cells matter. Your body has about 100 trillion human cells. But here's the twist: you carry even more bacterial cells than human ones. To understand cell culture, we must first understand what kinds of cells exist. A cell is the smallest unit of life. Some organisms are made of one cell. Others, like plants, animals, and humans, are made of trillions. What do bacteria, plants, animals, and humans all have in common? They are all made of cells, but not all cells are the same. Scientists group every living cell into two major types: prokaryotic cells, like bacteria, and eukaryotic cells, which include plants, fungi, animals, and humans. In simple terms, every living cell falls into one of two categories, either prokaryotic or eukaryotic. Understanding this difference is the foundation of cell biology and a key step toward mastering cell culture. Despite their many differences, prokaryotic and eukaryotic cells share several key features. Let's look at what they have in common. Both cell types contain DNA, which stores genetic information and controls how the cell grows and functions. Both have a cell membrane, a lipid barrier that protects the cell and regulates what enters and exits. Both rely on essential biomolecules, including proteins, carbohydrates, and fats, to build structures, produce energy, and carry out cellular processes. They also depend on minerals and vitamins to support enzyme activity, metabolism, and overall cell health. These shared features explain why basic biochemical principles apply to all cell culture, whether you're working with bacteria or mammalian cells. Understanding these similarities helps us design culture conditions that support fundamental cellular needs. Prokaryotic cells are ancient, dating back nearly 3.5 billion years, among the earliest forms of life on Earth. Eukaryotic cells appeared much later, around 1.5 billion years ago, marking a major step in cellular evolution. There is also a clear size difference. Eukaryotic cells are roughly 10 times larger than prokaryotic cells. This difference in age and size reflects growing biological complexity over time. Now let's look at the most important difference between prokaryotic and eukaryotic cells. Prokaryotic cells do not have a nucleus, do not have membrane-bound organelles, and their DNA floats freely inside the cell. Eukaryotic cells, on the other hand, do have a nucleus, and contain specialized organelles like the mitochondria, endoplasmic reticulum, and Golgi apparatus. The name itself explains this difference. Eukaryote means true nucleus. Prokaryote means before nucleus. This structural difference affects how cells function, how they produce energy, and how they are maintained in the laboratory. Now let's connect this knowledge to cell culture. Bacterial cultures involve prokaryotic cells. They grow fast, require simple media, and are easier to maintain. Mammalian and plant cultures involve eukaryotic cells. These require strict sterile conditions, specific growth factors, and carefully controlled temperature and carbon dioxide levels. The key takeaway is simple: the type of cell determines how we culture it. Understanding the cell type helps scientists choose the right conditions, tools, and protocols. One effective way to understand cell structure is by building a physical model. For example, a plastic bag can represent the cell membrane, Jell-O can represent the cytoplasm, spaghetti can represent DNA, Cheerios can represent ribosomes, and a rubber band can represent a flagellum. This activity helps connect cell structure to cell function, making abstract concepts more visual and memorable. Let's take a moment to compare. Can you list 3 similarities and 3 differences between prokaryotic and eukaryotic cells? Remember the similarities: DNA, a cell membrane, and ribosomes. And the differences: the presence of a nucleus, membrane-bound organelles, and cell size. This quick reflection reinforces your understanding of both cell types. Understanding prokaryotic and eukaryotic cells is the foundation of cell culture and modern biological research. In our next video, we'll explore how cells are grown in the lab, including culture media, growth conditions, and essential lab practices. This video is part of the MusB Research Cell Culture series, Bridging Biology to Real Science. If you found this video helpful, please like, share, and subscribe to MusB Research. Turn on the notification bell to stay updated on upcoming episodes in our cell culture series. And let us know in the comments what topics you'd like to see next.